All right, well, good morning. Welcome back. I hope that you all were able to find the link for the video recording for Monday. It's on our Canvas site. Let me know if you have any problems with it. Otherwise, we did have the midterm due, but I had a couple of people email me saying they could not access it. So I'm going to open it back up for those that could not access it. And we'll go over that once everybody finishes and takes it. And I'll just set that as a due date. Let's just say Friday evening. That way everybody's got a little bit of extra time. Anyway, we'll get back on to second part of chapter 9 here, looking at the propagation of plants. And here we go. So talked about genetics on Monday, and we're going to get into propagation of plants. So forgive me if I kind of move through this a little bit quickly. We left off talking about kind of the asexual propagation and how we have a lot of cultivated varieties of plants that have really been around for a long time, just existing as clones. In some cases, there's the Corinth grape variety that has been around for over 1,000 years, just because it's kept being propagated by cutting and grafting. So whenever we're doing asexual propagation, we're basically just cloning that same DNA and perpetuating it. So we'll look at some of these vegetative propagation methods today. Some are simple, some are more complicated. So if we're looking at cultivating a variety through seed propagation, we have to be very careful about the source of that seed. And the big thing that we're looking at is trying to prevent cross-pollination with other cultivated varieties and species. So in order to do that, what seed growers will do, whether it's a hybrid variety or an open pollinated variety, is maintain lines of those varieties and keep them isolated to prevent that cross-pollination, which then will result in a loss of genetic purity. And it depends on the plant, of course, as how it is isolated. You can either do that isolation in space, for instance, keeping one line separated from another by a certain number of, say, feet or distance, whatever, kilometers, miles in some cases, depending on how it's pollinated. So if it's wind pollinated, then wind can carry pollen potentially about a mile. So say corn, for instance, that is wind pollinated, we would need to keep varieties or the same species separated by about a mile if there are no significant geographic barriers. If you have barriers in there, then you can still have it isolated without getting that exact distance. And one good example of that is in corn would just be to have, say, a field of corn and only save seed from the plants that are on the very inside of the field, because the outside is likely to get cross-pollinated from other corn plants from maybe fields that are nearby, but the inside is only likely to be cross-pollinated by the plants that are closer to it. In corn, there's an interesting thing that we can look at as a way to ensure that. And it's the fact that corn is one of these few plants that has the effect known as xenia, or jumping genes, where when one corn pollinates another, it will actually move some of those characteristics, such as color. So say you have a yellow corn and a white corn. The yellow corn pollen lands on the white corn's silk, and then it pollinates that kernel. It's going to turn the color of that kernel yellow. Also, if you're doing, say, like a starchy corn versus a sweet corn, if that starch corn pollinates it, then the sweet corn, then it'll make it more starchy instead of sweet. there are only a few plants that actually exhibit those jumping genes, as we kind of mentioned earlier on. But that's one way to look at isolation. Another way to look at isolating a line is to do it in time. So say you're planting sweet corn and field corn, and you have like a 60-day sweet corn versus a 100-day field corn. They're not likely to be flowering at the same times, so you could still save seed from both of them and isolate each of them in time, because that cross-pollination is not likely to happen. There is some variability that can be tolerated, or in some cases even desirable in some of those cases. And one instance might be just maintaining an open pollinated line. For hybrids, we can't have that because we need to have the two lines crossing each other to get the reliable results. For an open pollinated line, one that we're not kind of preventing that cross-pollination of, they've become stable varieties, which they usually take to about the 5th generation, selecting for the particular traits to keep them stable. So when you save seed from them, they're likely to be very similar to the parents, although not exactly the same. And in that way, we can also get some traits crossing over that might be desirable. And it allows it to actually, adapt and evolve a little bit. Some of this we're just repeating here. So the seed certification programs we already talked about. So pardon me as I skip over this. And we talked about scarification, stratification, double dormancy, seed germination. Sorry, guys. Had some overlap. All right, here we go into the vegetative propagation. So when we're doing vegetative propagation, we're looking at it through the process of mitosis, which is just the non-reductive cell division process by which a plant is regularly growing. Just the splitting of the chromosomes, cloning themselves, basically, where your chromosomes are not changing. And what mitotic cell division can do is produce adventitious shoots and roots, as well as callus cells, which form as a result from cutting or wounding of particular cells. This is necessary for successful vegetative propagation. And that word adventitious just means that it's coming from a meristematic region other than what you would expect. So for instance, if it's producing adventitious shoots, then it's probably coming from roots. You're not expecting roots to form shoot cells, but in a root cutting case, it can do that. On the other hand, if we have adventitious roots forming on the above ground parts of the plant coming from, say, stem or leaf cells, then those are adventitious because they're coming from an area other than the meristematic region of the roots. So here's just an example of that. You can see on this stem here, we've got roots forming. That looks like a forsythia cutting there. Actually, this is from a root cutting, so we're getting the shoot forming once it's put in a different environment. Same with the roots. This is a cutting taken from a stem, and we're getting root tips coming from that stem cell. And you can actually kind of see right here, that's where it was wounded, and where that callus, that parenchyma cell is created, then the roots form as a result from that. A callus is just a mass of undifferentiated proliferating cells known as the parenchyma cells. And this callus tissue forms as a response to wounding. So here it is in the stem of a grapevine. And we will actually do some wounding in order to have the callus, because the callus creates those undifferentiated cells. Then once it has that proper load of the correct hormones, the ratio of auxin to cytokinin, it will also produce roots. So vegetative propagation we usually use in things that do not breed true from seed, that are heterozygous, having differing traits. In some cases, we need to completely avoid the use of seed altogether. In some cases, the hybrids will not even create sterile seeds, so they have to be vegetatively propagated. So we're going to talk just for a second here about grafting, where we will actually take two different vegetative parts of a plant and combine them to become one plant. So the two parts are the scion, which is the above ground part, and the rootstock, which is the below ground part. And a lot of our fruit trees we reproduce by grafting, apples for instance, because most apples are cross-pollinating via insects. Insects can carry pollen potentially several miles. So when you get seed from apples and you plant it, it's not going to be true to type. It's going to be totally different than either of the parents. So we'll take a cutting of the apple and we'll place it on a rootstock that performs well. And we get the genetics of both. We kind of get the best of both worlds in that case. We're getting a strong, vigorous rootstock that's probably disease resistant, as well as the top growth that we want. So the flavor of a particular apple variety, for instance. Almost all of our fruit and nut cultivars and many of our ornamentals are clones, which basically just looking at those as an extension, a perpetuation of the original stock plants. So they all have the same genetic makeup, the genotype, and can potentially be exactly alike. However, we have to think about the phenotype as well, how the environment turns the different genes on or off. And so it's not, they don't always look exactly the same, even though they have the same genetics. And we look at cultivated clones as originating in two different ways. First, as seedling plants that are propagated, just that have different genetics, and that should say sexually, not vegetatively. Sorry about that. Or we can also have them arising from mutations. And mutations, as we talked about, can occur naturally or can be promoted by the use of chemicals or radiation, or in some cases even just severe mechanical or physical treatments. Either way, it starts as a single cell that has different genetic makeup, and then arises, continues to grow, developing that genetic makeup. So in a lot of trees, we call this sports or bud sports, where just one branch will grow differently than the rest of the plant. And we would take that branch and then propagate it. Some of these mutations will change genetically only on a portion rather than the entire shoot. And in this case, we call them chimeras, like the mythological creature. So in this case, we've got a chimera, a variegated pink lemon known as the Eureka lemon, which is a type of chimera because it actually has both types of genetics in it. So we get this variegated foliage as well as the variegation in the rind of the fruit as well. So chimeras can create some negative effects as well as some desirable traits. And a lot of fruit and ornamental crops, especially the variegated ones, started as chimeras. Here's just a look at the different type of chimeric plants, which are, in this case, being categorized as where those mutated cells occur and kind of how they develop. So we have a mutated cell occurring in a specific spot and will sometimes just happen in one region. And so in this case, it's a mericlinal chimera, where it's just only on this one part that we have that one mutated cell and it's isolated to that particular region. A periclinal chimera is happening kind of at that meristematic region where it's moving throughout the entire growth plate. So that's a periclinal chimera. Those are more reliable to develop into a mutation that you could then propagate and perpetuate. Sectorial, again, it's kind of similar as the mericlinal, except instead of just staying on a region of the growth plate, it grows into a section possibly of the meristem as well, but it's only going to be on one section. So for our purposes, the periclinal chimeras are the easiest to propagate and perpetuate. It's kind of what we want to look for because they're relatively stable due to their positioning near that apical dome where that meristematic region is and those cells are being differentiated. So for the mericlinal and sectorial chimeras, we might get shoots or leaves that are not chimeric. So we don't necessarily get reliable propagation, although we can every once in a while get lucky. One thing that we have to look at in clones is, if we're taking it, it is taking that exact same DNA. So if it's affected by a disease, you're going to be carrying that disease over. Also, they have the same genetics. So if we have a disease affecting them, then because they have the same genetics, they're not likely to be able to adapt, and probably all the clones will die. There's the classic example of this is in the Irish potato. European settlers came to South America. They sent, one type of potato back to Europe. And then in Ireland, it became a staple crop. But then they're all growing that same, it's a genetic clone because of the way we propagate potatoes. We're basically cloning them. So when the potato blight hit, it wiped them all out, leading to a famine. We can remove some disease-causing viruses from the clonal material by heat treatment or by using shoot tip culture, where we're growing it on an aseptic environment in a sterile medium, often in a lab, where we're basically eliminating any viruses or diseases. And now there are some certification programs for that propagative material. All right, let's talk about cuttings for a minute here. A cutting is just a piece of vegetative tissue that is going to regenerate whatever part is missing, whether it's a root, shoot, or both. And we look at the cuttings being classified as to where they are coming from. So we've got stem cuttings, guess what, coming from the stem, which are going to have both terminal and axillary buds, but they have to form new roots. Leaf cuttings have no buds or roots, so they have to have the capability to form both adventitious shoots and roots. Leaf bud cuttings, just like leaf cuttings, except they have a bud. Root cuttings, taking a cutting of the root, so it needs to develop an adventitious shoot. So here are just some examples of different types of cuttings. On A here, we've got a hardwood stem cutting. That would be taken when the plant is dormant after a season's growth. B, we've got a stem cutting that has some leaf tissue attached. C, we've got a leaf cutting, so we're taking the leaf itself, and it's got to develop both an adventitious shoot as well as adventitious roots. D, we've got the leaf, but it's attached to a bud, taking out a node there, so it's easier for it to produce the shoot and root. And then E, we've got a root cutting, so we're actually digging up a root. It's got to form that adventitious shoot. Back to grafting. Basically, we're looking at it as joining these different parts of the plant together so they will unite and grow as one. The scion being the upper part, the rootstock being the lower part. And we get the best of both worlds. We can do both shoot grafting as well as root grafting. So root grafting, we're basically just looking at grafting the bud, the scion, onto just a piece of root. Whereas top grafting or top working, where we would be changing the top of the plant. So we would cut off the top and put the scion inserted into the cambium section to change the cultivar on a tree. We also have bridge grafting or in-arching, which is a type of grafting that is used to basically repair damaged tissue on a trunk usually, or to replace an injured root system. Whip grafting is the most common and most simple form of grafting used, where we use smaller diameter stems, and basically we're going to make a sloping cut about 45 degrees on both the rootstock and the scion, and then we're also going to make a second downward cut, which creates the width and the tongue, so that you have a little notch to where they fit together and are held together a little bit easier. And generally what we would do is, as we're, once we've prepared both the stock and the scion, we would then unite them and tape them together so that callus can form and can heal over, and then the top grows as the budding part of the plant, and the rootstock is, of course, the root part of the plant. It's better with smaller material. We're going to make those cuts to where they are interlocking, and then we're going to either tie it or tape it, and sometimes wax it as well, until that callus forms. And basically what we're doing here, one, keeping it united, keeping it closed to where it's those cambium cells are in contact with each other and can transport water and nutrients up through both of those stems until that callus forms, keeping it moist enough, preventing pests and disease from getting in. Cleft grafting is used for top working, and you can use a specialized tool as they have put here, or you can also use things like a meat cleaver even, or a hatchet, to basically get a split in that tissue and opening up that cambium tissue on the edge. And then we would take scion wood and kind of creating it at a wedge, ideally the same size, the same size as the cut that you've made in the rootstock, and then sliding that in there to where you get that vascular cambium contact. The big thing in grafting, no matter what form of grafting it is that you have the cambium layer in contact. Because remember, that's the living part of the tissue, and that's where it's creating new xylem and phloem. So here's continuing on with that cleft grafting. With this cleft grafting tool, we use that tip to keep it open until we can insert the scion wood into these different areas of the stock. Here they are inserted. We have to be sure, of course, that the buds are pointing upward. If they're pointing downward, it's not going to work. And it needs to be done with dormant buds. So many types of grafting are done when the plant is dormant. So usually late winter, early spring, until we get some growth. We don't want to do it in the middle of winter because it's often too cold and doesn't allow that callus to grow back as easily. For top working and cleft grafting, we usually put 2 scions in there so it's matching up on both sides. And then we'll remove one of those to kind of promote the central leader.
Can I ask a question?
Yes, please.
Excuse me. Why does it need to be like dormant? Just so it's not in growth mode, so it can more focus its energies or whatever?
Oh, yeah, exactly. So, and there are some forms of grafting that we can do when they're not dormant, when they're actually growing, specifically budding. But the whole thing is where the plant is putting its energy. And if they're already, especially the scion wood is the most important part. Because the rootstock, you know, you're cutting it off, so you're kind of limiting the flow to where you're putting the scion wood, the flow of energy. But if your scion wood, the buds are not dormant, then they will basically, you know, not put the energy into developing the callus. They'll put it into actually opening those buds, opening those leaves. And when the leaves are open, it's sucking that moisture through transpiration out of it. So it's not getting the moisture into, you know, developing those callus cells. Does that make sense?
Yeah, it makes sense. I just wanted to make sure. Cool.
In this case, they're waxing the covering, completely covering it to prevent it from drying out. And you can tell they've used some scion wood here that's not the tip of the plant. If you're using the tip, you don't have to wax the top. But if you have an opening at the top, which, you know, we can get several sections of scion wood from one branch potentially. So if you do that, then you need to also wax the top of it just to prevent it from drying out. And that's an excellent question, Tom, because, you know, you can theoretically be successful as long as your scion wood is still dormant. So what a lot of people will do is, you know, say for fruit trees right now, when they're able to see the characteristics of the fruit tree, they'll tag them. They'll come back later on and take the scion wood, and then they'll store it until you have the ideal time for grafting. You can actually purchase scion wood online, or in some cases we can trade it. There are actually groups, you know, online. North American Fruit Grower Exchange, NAFEX, is one of these groups where they are trading scion wood. They'll send you, in some cases, just cuttings or scion or buds, and then, you know, you can use that to do your grafting. So mentioning budding is widely used also in fruit trees and especially in roses. And tea budding is the most common type of budding technique. And the reason it's called tea budding is that we're inserting these buds through a T-shaped cut in the bark of the woody plant that we're putting it on. And then we're tying those buds in place or taping them. More often these days we're using more tape than tying because it's less constrictive and holds in the moisture a little bit better. We're talking about mostly doing woody plants right now, but you can do herbaceous plants you can graft as well. And there's been a lot of development of the science on grafting herbaceous plants, especially in things like tomatoes. Especially some heirloom varieties where we want that flavor, but they don't have the disease resistance. So they'll put disease-resistant rootstocks, plant those, and then they'll use the same techniques that we're talking about to graft them on. The big difference here is that they're not going to be dormant because they're annuals and they don't go dormant. So you have to basically really be sure to keep it very moist so it doesn't dry out. They're more prone to drying out because they don't have that protective layer of the bark. So in the grafting, we want to be careful to promote the healing. And the big reason why we're doing it when the plants are dormant, so that the union where that scion and rootstock are meeting can heal and form a callus covering it so that it unites as one. And this usually takes about 2 weeks. And we need to do that again before the buds on the scion start to grow and develop, because those transpiring leaves will desiccate the scion wood. and prevent it from healing over. So here's the cleft graft, looking at it from the top. And we put it on the outside so that bark layer is matching up. And think of this line right here as that cambium layer. That's the most important thing, because that's where the healing is going to occur. And that cambium matching up is going to allow for the nutrients and moisture to be transported from the rootstock to the xylem. So this is, basically just looking at an image of those parenchyma cells beginning to unite. So here's the callus forming kind of in between and interlocking, so they're growing as one. And then beginning to differentiate and develop the different new xylem and phloem tissues. There's the new xylem and phloem formed, which is going to permit that passage of those nutrients and water into the xylem wood. So there is some limit to doing grafting, and ideally we want them to be closely related botanically. The closer they're related, the better. So if they're in the same genus but different species, then you can still have some success. But even with very close relationships, it doesn't always work. I usually say that go into grafting thinking that about 50% of them are going to take. That way you don't get disappointed, and then you're pleasantly surprised when more of them do take. If they're both clones and you did a good job on the grafting and matching up the tissue, then it's more likely to get closer to 100% success. Some cases we can actually look at intergeneric combinations. And it just depends. I mean, there's a lot of research out there done on this already, and a lot of it that's not been done. But I'm sure you guys have seen some cases where we have multiple fruits growing on one tree, like the Burgess catalogs that you get in the mail every once in a while. They've got the 3-in-1 fruit tree. So you could get, for instance, pears and apples, multiple varieties on the same tree through this intergeneric combination, because we've got two different genera there. We've got the apple, the Malus genus, the pear being the Pyrus genus, or as they mentioned here, pear and quince. So if you graft pear cultivars onto quince roots, some of them will be successful. But if you do quince on pear roots, for some reason, they always fail. We've actually been doing this quite a bit here with the calorie pear, which is kind of growing like a weed around here, but it's a really strong root stock. So we've been trying to see if we can graft apples onto it, and we've actually had quite a bit of success in doing that, an intergeneric combination. So those happen. Plums, for instance, can be successfully grafted on peach roots, but peaches, for whatever reason, don't work on plum roots. So, however, those are fairly closely related. You need to be at least in the same family for grafting to work. You can't do it outside of families. And all these plants that we're talking about right now are in the rose family. So over here on the right, we've got an example of a locust graft. And we're going to have this, usually we see some sort of swelling point on grafted plants. That's where that callus tissue combined. And that is kind of a weak point. So it's better to have that closer to the ground. But in this case, we got a purple flowered locust planted onto a regular white flowered rootstock, or grafted onto it. You can see that swelling point there where it was grafted. Often what tree fruit growers will do is looking at having harvesting easier by grafting onto dwarf rootstocks. So that dwarfing genetics comes from the roots rather than the scion. So we have dwarf or semi-dwarf rootstocks that we're grafting on the varieties that we want to get the particular characteristics of the fruit, which is fairly interesting. In some cases, the rootstock can strongly influence the quality of the fruit as well. So you have to be careful about selecting both the scion and the rootstock. So now we're, as I mentioned earlier, we're starting to see more herbaceous plants being grafted. And that's mostly as a result of the ban of methyl bromide as a soil sterilant fumigant. That was a way of disease control, but it was found that that chemical does a lot of damage to other organisms. So they banned it. So now to get disease resistance, they're finding disease resistant rootstocks and, you know, grafting the ideal version onto those rootstocks. Layering is another form of asexual propagation in which the part of the plant that we're trying to get a new propagule from, a new plant from, is still connected to the mother plant. And usually this is a very successful way of asexual propagation because they're still united. Up here we got an example of tip layering. This only works on a few types of plants. Raspberries, some roses will do it, where you're just burying that tip and it will grow a new shoot. And in doing so, it's also growing new roots. Mound layering is often used in the production of rootstock for fruit trees. So you find the ideal rootstock, you actually will top it and then cover it with a mound of soil or media. And then as it produces new shoots, it's also going to produce new roots. You uncover it, snip those back, and you've got your rootstock. Here's simple layering, where basically we're just bending part of it, putting it underground. We'll often put a little cut or notch in it there so that you have the auxin and cytokinin kind of accumulating there, and you have the wounding, which develops that callus tissues that roots can develop from. But you leave the tip above ground. So it's still connected, still getting the nutrients and moisture from the mother plant, but it's developing a new one there. So there's simple layering. And then here's an example of air layering, where basically we're going to remove that bark tissue, expose that, cambium. Then we're going to cover it in some sort of media, often like a moss. And we're going to tie that media, cover it onto that branch. And then we like to use like a plastic wrap so that you can see the roots forming. When you start to see those roots forming, you just snip that off and you got a new plant. Describing the process there. You do have to keep that layer moist though. So in the greenhouse, when we do that, we also try to water those layers. Some plants are going to have these specialized structures, usually things like, in this case, the runners of the strawberry, also known botanically as stolons, where they're producing these baby plants we call plantlets on these runners, and all you have to do is just detach that from the parent plant and you got a new plant. It's a very simple, easy form of asexual propagation. We can also use suckers in some cases, especially in like blackberries and raspberries. They're going to develop adventitious shoots from a horizontal, underground root system. So basically we're dividing that. We're just going to basically stick our spade or our digging fork in there and pull it apart from the rest of the plant. As long as there's a shoot and some root tissue, it will grow and develop into a new plant. We can also look at doing division from the crowns of the plant themselves. So most perennial plants are existing as this single unit, and that crown right here is developing from the base. And as it grows wider each year, you've probably seen this before, some perennials will kind of flop over in the middle. We can cut pieces of that crown off, and as long as it's got shoots and roots, it will develop into a new plant. We can also look at using the other specialized structures in plants, such as the bulbs of flowering things like daffodils, tulips, corms like in gladiolus, tubers like in potatoes, tuberous roots like in dahlias, rhizomes like in bamboo. These specialized structures often are developing both adventitious shoots and roots anyway. So all we have to do is divide them into a section that has a bud, pretty much, and ideally has some roots connected, and they will grow into a new plant. We can look at this as separation or division, and the big difference here is that in separation, we're utilizing the natural form of the plant's response. So in a lot of these bulbs, for instance, they will develop a little baby bulb at the base of that bulb. So you just go in there, you dig the bulb up, you pull off that baby bulb and you replant it. That's separation. Or garlic, for instance, is an example of separation. We're growing that clove. You know, it's got these multiple bulbs in there. We're pulling them apart, those different cloves, and replanting them. Very simple form of separation on a commercial scale. If we have to actually physically cut it apart from the crown, then we refer to it as division. So a bulb is just a short underground, basically, plate of stem tissue that has these fleshy scales surrounding it. And we have three different types of bulbs: a tunicate bulb, a scaly bulb, and a non-tunicate bulb. And those are just slight differences in the way that they are propagated, whether you have to pull the tunic off or not. A corm is also a swollen underground base, but it's more of a like horizontal. So just a few different flowers in that, gladiolus, freesia, and crocus. And they develop new corms just above the old corm. Sometimes they will grow into new plants, sometimes they won't. We can just pull those apart and replant them to get a new plant. For tubers, you know, the common tuber that we think of is your potato, right? So it's cut into different sections where we have different buds that we call the eyes on the piece. That becomes the food supply for the developing shoot until it develops new roots. Another commercial form of division in that case. And all of our potato cultivars are clones and are propagated through dividing them into these several different sections. Tuberous roots are just root tissues that develop almost like a tuber-like section. Sweet potatoes, we look at developing them, although in this case we develop them into slips. So usually what we'll do is we'll take a section of sweet potato and we'll plant it in some really rich media, bring it into a warmer environment and get it to form the new shoots. The new shoots root readily when they're put into water. So we can just pull those shoots off, stick them in water, they root, and then we can use those as the sweet potato slips. Rhizomes are horizontally growing stems, basically usually just right below the soil surface. In this example, we've got bamboo. So all you have to do is just take a section, cut it where you've got both some root and some bud, and then it will develop into a new plant. And then some other plants have, you know, different specialized structures. I think the big thing with, you know, developing plants from these structures is just to look at having some bud tissue on it and ideally some roots connected. So we could look at bananas, ferns, ginger, many different grasses. These are economic plants that are propagated from rhizomes. And in many cases, that's how they're sold. You purchase a rhizome of these plants, bare root, that's what they send you. We also have the above ground horizontal stems, the stolons. They are propagated the same way as the rhizomes, just separated where you have some root and shoot tissue. The last form of propagation we'll talk about here today, and the more advanced form, is micropropagation or tissue culture, where we're growing pieces of a plant known as explants. That can be as simple as just one cell of a plant. Often it's just a little section of meristematic tissue, whether that's in the shoot or root. And then we use those, we put them on some sort of aseptic culture, some sterile media, and we give them everything that they need in that culture to grow them into new plants. And the cool thing about micropropagation is we can make, many different, we can make thousands of new plants just from a few pieces of tissue. In some cases, we can use seeds as well to grow on aseptic culture. We can extract embryos and grow them. We can do shoot tip culture. Either way, we're going to excise some form of that meristematic tissue, whether it's stem tissue or root tissue, and place it on this nutrient medium. And then it's going to develop the callus masses just through its regular cell division, because that nutrient medium is giving it the nutrients in order to develop. We also use this, though, that media is going to have favorable growth for bacteria, fungi, and yeast, so we have to sterilize it. Both everything that we're handling, even our hands, we have to cover them, because we can introduce even just one cell of a bacteria or fungi, and it will colonize that media. So we got to sterilize everything. That's why it's called aseptic culture, and why it's usually done in a laboratory. And then we're going to give it everything that it needs: the mineral salts, the vitamins, the sugar, and then the growth regulator. So we're also providing the hormones. And in some cases, it takes longer to develop new plantlets. So in those cases, we have to sometimes reintroduce new nutrient media. So we use things like coconut milk, yeast extract, and banana puree as a way to get those nutrients in there. This is just a chart looking at the different ingredients in the media to the development of the plants. And so again, this hasn't been done on all plants, but there are often particular formulas of this media that they use for particular types of plants. The plants that are already commercially propagated, they already have these directions out there. Some of them, you just have to experiment with. When we start to get them to develop into the new cells, we have to introduce the cytokinin and auxins at different levels to get them to produce new shoots and new roots as well. So it's a complicated process, but it can even be done in a kitchen environment, as long as you have the sterilized media and the sterilized tools that you're using. Micropropagation can be done. All right, so that's it for propagation. Anybody have any questions, comments, or concerns? So I'm going to post a little bit of a kind of a take-home lab for this as well. And basically, it's going to be very open-ended. It's just trying to get you guys to propagate a plant at home. And I'd like for you guys I'm going to make a little discussion board so you can post your results. And I'll talk a little bit more about it on Monday. But be looking for things that are developing adventitious roots forming above ground. Those are the easiest to propagate, and it'd be fun to just do something that can be very successful. But email me if you want any more information on any of this, or send me a message through Canvas. If you want to try out micropropagation, I'd be happy to provide you with some directions on how to do that, even in your own home.
Cool.
All right. Well, for those of you that didn't already finish it, get on there and finish up the midterm exam, and we'll just have that close again on Friday so we can talk about it on Monday. Make sure everybody's got everything they need out of it. Otherwise, you all have a great day.
Okay. Is Hunter there?
Hunter is here.
Okay. Well, I'll be calling back in a couple minutes then.
Thank you, sir.
Okay. Bye, everybody.